So it's recording right now. Yeah. So now that exam one's over, you know, finally, at least you got through your first biochemistry exam. We're gonna start going through exam two material. And the thing that you should be aware about exam two is that it's a lot of content. And it's not like it's that you can kind of understand it's like straight memorization, okay? Because it's a lot of chemical pathways. So the one thing I want to say right now is that start studying for exam two as fast as possible. Okay, so you're gonna notice that in my exam two folder in that shared Google Drive that we have, I already put in a worksheet that one of my really good friends who was an SI for over a year, she made a really good worksheet that's basically 3 pages of all the reactions you need to know for the nitrogen metabolism part, because that's like the big part of this peerage section of the exam. So as you go through those lectures, please start filling that worksheet out, because that's going to be a great study guide for you guys, because it's all the information that's hard to memorize condensed into one little packet. Okay, so it's
So this
already
is not just what's been done so far. This is the whole exam stuff.
for that worksheet is going to be only for the nitrogen metabolism. Yeah, that the one that you have right there, that's going to be just for the nitrogen metabolism for the most part. But exam two is both Dr. Purich's materials and Dr. Xylie's materials, and they teach completely different things. So right now that worksheet you had, that's going to be mainly corresponding to Dr. Purich's materials. Okay, because Dr. Xylie's material isn't necessarily memorization, it's more like a review of like the plasma membrane and stuff like that. So it's a little bit easier, but like Dr. Pierce stuff is a lot of reactions, right? So just to start, I just want to have a little reflection time. So how did everybody feel after their first exam? Any okay, Corey is not.
I got like halfway through and I was like, yeah, this is not good.
I mean,
I was seriously considering dropping the course.
but at least you got like that first experience of how it is, because the questions are sometimes confusing, right?
Yeah.
So you like every class that I have, I usually do the worst on the first exam.
Right.
Because I never know what to expect. But now you know what kind of to expect, right? So that's a good thing too.
Yes, and I did do fine. I was overreacting, but it was.
I mean, it happens. I think every time I finished the biochemistry exam, I would just go to Chipotle and get like the biggest bowl and chips just because I was just exhausted and stressed. Matt, you were gonna say something?
Yeah, can I ask a stupid question? I've never had a curved exam before, and so when she said the average was a 72 plus or minus 17, what does that mean? Does that mean like if you got a 100 on the exam, that you actually got an 83?
So
And
if you want to
like,
ask some of
what's
these
the
more
score?
detailed grading
Yeah,
questions, I would just email Dr. Duma. But if you want to refer like to the standard deviation, it's just like a bell curve. Like standard deviations are separating percentages of people like who got above or below a certain grade point, if that makes sense.
you know what that's plus or minus 17 means? That's
Yeah, it's like
the
standard deviate. Yeah. It's like another section of like that middle, and then you can have even another one. But yeah, as you go towards the ends, the percentages get smaller. But I think that's what she meant. But I would just email Dr. Duma with these questions because she specifically told all the SIs like, if there's any grading questions, don't say anything because we don't deal with the grades. She manipulates them. Yeah. Okay, but anybody else want to talk about their experience with the exam? Good, bad, mediocre?
Um, so I feel like I was pretty happy with my grade, but that actually worried me because I heard that this one was gonna be like one of the easier ones.
I mean,
So does it only go down from here?
I mean, the thing is, it does go down, but the averages go down too. So it's usually balances everything out to an extent. So like as I said, like usually for the cutoff for like an A in biochemistry for the undergrads, I don't know if the grads has a significant difference, but I doubt it's that big. Usually the cutoff for an A is an 84 towards the end. So it's usually gonna be around that area, and you're gonna see that the biggest drop in your average, and that's why I'm telling you guys right now about exam two, is exam two is the biggest drop in the average. Because most people get a little bit complacent, a little bit like, oh, I did okay in exam one. They just don't like exam two is a hard exam, but it gets a lot better after exam two. Exam two is the huge dip, but exam three and exam four is a lot better.
well, that's good to know. Thank you.
Too, don't run away crying.
I'm giving you multiple warnings about this exam. Like I'm telling you guys, start studying for this one. But just moving on. So we
This one's the worst because the TAs last week said they just get worse from here on out. So if
I think that's an exaggeration. I think that's a little bit of an exaggeration, but I do agree that exam 2 is the hardest one.
okay, good to know.
Yeah.
Thanks for the warning.
Dr. Duma said the same thing too, that the first exam usually has the highest average and they go down from here on.
Yeah, but like exam 2 is a huge dip, and then exam 3 goes back up, and exam 4 is stable. That's how I see all the exams, like from last semester. That's how I felt that I saw the grades, because exam 1 is usually the easiest one, just because you have to admit, like the first 3 lectures are pretty simple for the most part. It's just when you get to the math parts that get super complicated. But like you guys are definitely gonna be fine. Because I think when you say it definitely gets worse, it implies that exam 3 is gonna be worse than exam 2, and then exam 4 is gonna be. I don't feel like it's gonna be that. Exam 2 is the hardest one. But again, it's all opinion based. But just go moving on, just so we don't not waste time, but and have no time. I will be also posting a little survey in the group me's, just because I want to see if there's anything you guys want me to change in my teaching style, because every group is a little bit different. So I just want to make sure that you guys are getting the best quality educational reviews that I can offer. Okay, so I will be posting. I actually forgot to put an announcement page, but I will be posting a little announcement later on what the next slides are going to be, and I apologize for that. I don't know why, but this one slide didn't have it, but I will be posting a little announcement as well in the group me. But just jumping into lecture 13. So lecture 13 is our first overview of what metabolism is. And metabolism is going to be really, really important for the rest of biochemistry, okay? So metabolism is the totality of cellular processes that make and degrade chemical substances, okay? So this is what fuels vital processes. This is what is moving us, right? This is what's making us alive, okay? Can anybody tell me what anabolism and catabolism is?
Anabolism is building and catabolism is tearing down.
Yeah, exactly. So these are just two like separate subtypes of like metabolism, right? So anabolism is building things up, right? We build like fibers, we build tissue, and catabolism is just the opposite, right? It's breaking those down for its like building blocks, right? So, and enzymes organize themselves in multiple metabolic pathways, right? And you're going to see that a metabolic pathway that looks like this is going to have multiple enzymes, okay? And these enzymes, what they do is that they rely on these substrate specificities to be very specific in their metabolic pathways. They can actually form these supramolecular complexes where you're going to see like, it's almost like a quaternary structure, but on steroids. It's like so many enzymes close together, so the metabolic pathway is moving from one to the other without like having any issues of like the substrate getting lost somewhere or getting degraded. And they're usually in the same compartment of a cell, right? So they can usually be only in the cytosol, or they can be inside of mitochondria. So because of they're in this close proximity, this prevents issues that include substrates getting broken down when we don't need them and stuff like that, right? So the important thing about these metabolic pathways is you're always going to see a committed step. And this is the most favorable thermodynamic step in the whole reaction, okay? And you're going to see right here in this image, this is our thermodynamic, or is our favorable step right here. Everything else is reversible except enzyme 1. Enzyme 1 is pushing that reaction completely towards the product side, okay? So this is what we call our committed step, because this is the one area that we need to put a lot of focus to move that whole reaction forward. It's like that big push to push everything down, okay? But since it is such an important metabolic step, this is also the area that we have the most targeted. It's the most targeted area of our metabolic pathway, right? It's the target of allosteric feedback inhibition. So there's 3 types of regulations, right? We have our allosteric regulation, which is just a reversible binding in the allosteric site, right? We kind of looked at that in a non-competitive binding. So this is the allosteric regulation, right? And then we have a reversible covalent modification. So this is when a group is transferred to the enzyme. It's covalently binded to the enzyme. Then we have induction and repression, and these are going to be mainly gene regulations, right? So gene and mRNA controls that control the overall protein synthesis of our committed step enzyme. Okay, does that make sense for everybody? Any questions? Okay. So now let's look at the effects of an activator and the effects of an inhibitor, right? It's kind of self-explanatory what an activator and an inhibitor does, right? But it's also important to know, like, for example, that an activator is gonna increase the velocity of the enzyme, right? Of what the enzyme is doing to the substrate. And an activator can also lead to less substrates needed to reach the half E max, right? So our Km is decreasing, right? Because of this activator. For an inhibitor, it's just the complete opposite, right? We're going to decrease the velocity and then we're going to increase our Km, or in other words, the substrates needed to reach that half Vmax. Okay, and you see it in the graph right here. You see that an activator is decreasing the Km, right? As we see, the Km is the x-axis, and it's also increasing the reaction velocity, okay, and the y-axis. Okay, does that make sense? Yep. Right. Now we're just gonna jump into a little bit about bioenergetics, right? So this is the study of the energy, or the energy is transducing from process. So the energy that comes and goes into or comes out of metabolic pathways, right? So this is just the study of energy in a living organism. So again, we're just going back into our Gibbs free energy, right? We talked about this for exam 1. There are 2 new terms that you should be aware, or 3 or 4. Sorry, I just read all of them. So exergonic, endergonic, exothermic, and endothermic. Okay, so exergonic is just a favorable reaction. This is a negative delta G as an exergonic reaction. And endergonic is a positive delta G. Okay, and then when we're talking about enthalpy, an exothermic is a positive delta H, and an endothermic is a negative delta H. Okay. Yeah. Sorry, I said the complete opposite. Sorry. Exothermic is heat release, so it's a negative delta H because you're losing heat. And endothermic is when you have to put in heat, so that's why you have a positive. Sorry. And the important thing to know out of all of this, because he's definitely not going to go ask about Gibbs free energy again, because you talked about it in exam 1. But the important thing to know is that each biochemical reaction, each step in a metabolic pathway has its own delta G. Okay? And that's going to be really, really important when we're going to be looking at huge metabolic pathways. Something that I always get questions about is the difference between delta G and delta G prime. And I always like just to bring it up just because I know there's always going to be some questions about what's the difference. So the difference is that delta G prime is just the calculation of delta G under standard conditions of measurement. Okay. That's usually 1 molar, 1 atm, and 25 degrees Celsius. For delta G non-prime, it's just under non-standard conditions. Okay. Now, this might look kind of like a stupid question, but it's also pretty important to know, right? So I'm just gonna pick someone right in here. Let's see. Shelby. Sorry, I started you there, but are cells under standard conditions?
No.
Exactly. So cells are not under standard conditions, right? Because it's really hard to control conditions inside cells, right? Not every time it's going to be at a 1 molar of something at 25 degrees Celsius at 1 atm. It's hard, right? So that's why this delta G non-prime becomes really important in bioenergetics, okay? So again, this right here in the bottom is just a review of what we know about Gibbs free energy, like what's a forward reverse reaction equilibrium, right? So under here. Now we're going to start talking about ATP. And we know that ATP is really, really important for cellular processes, right? It usually is coupled with positive delta G reactions to make a negative delta G, because ATP is that good at moving things along. Okay, so we have these 2 equations for the hydrolysis of ATP. We have one that converts it to ADP, and then one that converts it to AMP. Okay, so Pierce doesn't really need you to memorize numbers, but I would just know that ATP to ADP is a negative 30 delta G, and then ATP to AMP is a negative 45. Okay, it just might be a little bit easier if you ask questions about certain bioenergetics. Okay, so I just always like to pinpoint those two numbers as you might want to know just in case. Okay, so why is ATP hydrolysis so favorable? We're going to go into a little bit more detail towards the end of Dr. Pirch's materials for exam 2, but just right now, it's really, really good at breaking itself apart because of there's a lot of high electrostatic repulsion. ATP has 3 phosphate groups, and these phosphate groups are very negative, and they hate being close to each other. So that's why it's very favorable to break it apart. Plus, their products are really, really stable. So they usually don't go back to becoming ATP. Okay. And here is just an overview of there's 3 different types of ATP hydrolysis. We have our phosphoryl group transfer, pyrophosphoryl group transfer, and our adenyl group transfer. Memorize these 3 types of ATP hydrolysis. Specifically, memorize where water is attacking. Is water attacking at the gamma phosphate? Is water attacking at the beta phosphate? Or is water attacking at the alpha phosphate? Okay, because these are all going to be there's usually one question about this types of hydrolysis, like which one is which. Okay, so the important thing to know is which phosphate is being attacked and what is your product. So if it's a phosphoryl group transfer, we know that water is going to attack the gamma phosphate group, giving us ADP and a single inorganic phosphate. Okay, but if our water is attacking our alpha phosphate group right here, right, we're actually going to be getting AMP and a pyrophosphate. Okay, so just be aware that you should memorize these reactions. Corey, you're going to say something?
Well, I mean, I look at that and maybe it's really obvious to other people, but how are pyrophosphoryl group transfer and adenylyl, how are their products different? Like how can we identify
And
how
why
those
is
are
that?
different?
Well, it's more of the way that they're breaking, right? So you're gonna see here that there's also a little triangle, and that's
Right.
representing the bonds that are being broken. So for our
Right.
pyrophosphoryl group transfer, the water's attacking this phosphate specifically and breaking it apart right here, giving
Uh-huh.
us the AMP and our pyrophosphate. In this case, the water's attacking. Sorry, the slide is getting right in the way. But the water is attacking our alpha phosphate group, but you're going to see that the bond is not on the right side of that of the alpha phosphate group, but it's on the left side. So we're still going to be with that AMP because the bond to the left of the alpha phosphate group is being broken apart, not to the right. Okay.
Okay,
It's
so
just
the
different.
products are the same. We're just concerned about where the bond is being broken.
Yeah. And which phosphate group is being attacked.
Okay.
So why does it matter? Like, why are we going to learn why one process would do one versus the other? Or like,
I mean, they're all going to give you really similar, like, the reason we were breaking ADP and ATP or ADP, for example, is that we want that energy that's given off once we break it. These are just different mechanisms that our body can break apart ATP. It's not necessarily that we're going to be in detail about why one is better than the other. It's just Purich wants you to be aware that there are different ways to break apart an ATP. David, you're going to say something?
It's that I think it's more so like, what's the best way to put it? The difference between 2 and 3 is more so where the oxygen from the water and the oxygen from the phosphates end up. Like it's the same product, but in 2 you can see the oxygen from water ends up on the pyrophosphate, where in 3, I'm not gonna say their names too long. 3 you can see the oxygen in the water ends up on the AMP.
Yep, that's actually a really good way of seeing it as well. I usually when I memorize just where the bond was being broken as well, but that's a really good way of seeing it as well.
Thank you.
So moving on to the next part of lecture 13, which is now where the ATP is going to come in, right? So this is the coupling of reactions, right? And you're going to see that this is the overall reaction to convert glutamate to glutamine, okay? But you're going to see that we're actually using ATP to reach this kind of transition state before we're able to move into our glutamine, right? So again, I kind of answered the question, but for glutamine synthesis, the reason this unfavorable reaction moves forward is because of ATP, because it pushes the reaction forward. It forces the reaction to go through because of ATP. Any questions on lecture 13 stuff? So now we're going to go to the roller coaster that is nitrogen metabolism. It's a lot of information that's just going to be thrown our way right now. So just jumping straight in. So lecture 14. So lecture 14, we're talking a little bit right at the beginning of amino acid, right? So this is a digestion and absorption of amino acids. So the important thing to know first is that the amino acids that we know of have a different form, and that's called a keto acid. Okay, so you're going to see that a keto acid of alanine is called pyruvate. Okay, and then a keto acid of aspartate is oxaloacetate. And you're going to see where these ketones are. Those are where our amino groups used to be, right? So these are like keto acids, just basically adding a ketone to our amino acid. So again, green asterisk means you have to have the structure memorized. So these three are going to be really, really important for the rest of biochemistry as well, these three keto acids. But there's three different sources for amino acids. We have digestion, so eating, right? Eating protein. Intracellular proteolysis, so this is when we break apart our own proteins to get the amino acids. And we have de novo synthesis, so which is when we can actually build our own amino acids, right? We don't need something coming from the outside like food. We can actually build it our own. But there are some amino acids that we can't build, right? So these are called our nutritionally essential amino acids. These are the ones that we need to get from our diet. And the best way that I think I really like this way that Dr. Pierce likes to memorize the private Tim Hill. So this is a really good way of memorizing these 6 amino acids that are nutritionally essential, right? So what is proteolysis? Anybody want to take a guess at it?
Breakdown of protein.
Exactly. It's a breakdown of protein. So this is how we break apart either our own proteins or proteins coming from our diet, right? So we have two very specific areas of our body that we usually associate with breaking apart a lot of protein. That's our stomach and our intestine, right? So in our stomach, we actually have pepsin, which cleaves after our phenylalanine, our leucine, and our glutamic acid. Then we have our intestines, which we have a lot of different enzymes, but 4 very important ones are chymotrypsin, which cleaves after tryptophan, tyrosine, and phenylalanine; trypsin, which cleaves after lysine and arginine; and then carboxypeptidase, which cleaves the C terminal of the amino acid individually. And we have our elastase, which just breaks apart elastin, right? Elastin is like important for like why our skin is elastic. So elastase is like present in a lot of tissue, right? But the important thing when we're talking about, especially for our stomach and our stomach, especially because pepsin is a huge zymogen in our body, is the concept of zymogens, right? So these are inactive proteases, so inactive proteins that break apart other proteins, and they're usually stored in the pancreas, and they only work when they reach a certain part of your body. In this case, I just want to be a little bit more specific with the small intestine because we are talking a lot about these chymotrypsin, trypsin, carboxypeptidase, and elastase, but the stomach does the same thing, right? So actually for the stomach, it's a little bit different, but for the intestines, for example, they go through the pancreatic duct into our small intestines, and based on the concentration or the pH of our, all the food and all the like the forgot the exact terminology for it, but all the like the all the food broken apart, depending on that pH, we actually activate certain zymogens. It's the same thing with pepsin, right? We know our stomach is really, really acidic, and pepsinogen, the zymogen of pepsin, is only activated at really low pHs, right? So this is just one way to make sure that we're only using these very dangerous proteins in the right location, right? Again, already answered that question again, but for example, we don't want pepsin to be activated out of nowhere in a different part of our body, because that's just going to break apart the proteins in the good proteins, the proteins that we want saved. So zymogens are just a little safety structure, right? to prevent these like bombs from going off until like they're in the right location to start. Okay. So then trypsinogen, which is the zymogen of trypsin. So there's different activations for trypsinogen. And in this case, they actually contain a trypsin inhibitor, but they're actually activated by an enterokinase, right? So this is just a different, it's basically a phosphorylation event in trypsinogen that allows trypsin to start working. Okay. Any questions on what zymogen is? The best way I like to think about it is just a way to prevent a literal time bomb from going off in your body. I saw a question. Someone was going to speak.
Yeah, so carboxypeptidase, it cleaves any amino acid at the C terminal.
Yeah. Carboxypeptidase is just cleaves right at the C terminal. So it's not very specific on which amino acid it targets.
Okay, gotcha.
Yeah.
Thank you.
So jumping in. So we know that our intestines have an epithelial wall filled with these microvilli, right? These like finger-like projections that break apart a lot of different nutrients, but they also start transporting it into our capillaries, okay? So amino acid absorption works with a symporter, okay? So you're gonna see that we have this symporter where we're actually bringing amino acid into our cell with the help of the sodium ion, okay? And again, this transport is driven by this ion gradient. But we need to make sure that we have a consistent amount of sodium in our lumen of our intestine. So that's where our sodium potassium ATPase comes in, right? So we're transporting 3 sodiums to the lumen and 2 potassiums inside using ATP. Okay, so we consistently make that ion gradient to allow this symporter reaction, this symporter transport to keep going. Okay. Any questions on this? So here it is, a little bit more about zymogens right here, right? So pepsinogen is kind of different from different zymogens because it's actually autocatalytic. So it can actually catalyze itself. So as soon as one pepsin is activated, it can activate another pepsinogen to become a pepsin and just keep going through that spiral, right? So it actually operates under low pH. As we know, the stomach has a really low pH. And it uses aspartic proteinase for catalysis, okay? But we also have different things called turnovers, right? So turnovers are periods of time inside our body where we want to start breaking apart proteins. And there's 2 different pathways that we can go through this. We have our lysosomal or phagosomal pathway. So this is when our certain organelles in our cell actually break apart other cells or break apart itself, and then a phagosome or a macrophage goes in there and just picks up what it can to recycle. And we have an ubiquitin dependent pathway, which is basically when we target a cell for death. Okay, kind of little savage there, but basically we put a little marker in that cell indicating to our immune system this cell needs to go. Okay. So these are different ways that we can do it. And we can do these pathways as well for proteins as well, specifically inside organelles as well. Okay. So these are 2 different pathways that our body basically targets cells or proteins to die. And the fun part, at least for exam 2, is there's going to be a lot of medically relevant material, right? Because it's kind of boring when there's no application. But for at least for exam 2, there's a lot of application questions. Take that as you will, if you hate applications or not. But there's 2 different medical conditions that Dr. Pierre wants you guys to know is marasmus and kwashiorkor. I hate saying that last one, always hurts my mouth. So marasmus is just basically malnutrition, right? So it's basically you're not getting adequate macromolecules that you need to survive. So this is showcased with extensive tissue and muscle wasting. And this is caused by inadequate intake of proteins and calories. So this is what we associate with famines, right? Kwashiorkor is a little bit different. So kwashiorkor means that you still have an adequate calorie intake, but you have a protein malnutrition, okay? So these have very specific signs and symptoms that we don't really see with famine, right? So these signs are irritability, enlarged liver, edema, and osmotic imbalance. I can talk a little bit about irritability because it's pretty interesting, right? So a lot of our proteins are also can also be converted into neurotransmitters, right? Neurotransmitters are really, really important for the connections between different neurons in our brain. So if you have an inadequate protein diet, you're going to have a very low amount of neurotransmitters and different hormonal signalings. That's going to affect a lot of your behavioral issues as well. Okay, so that's pretty interesting too. And then edema is just, fluid buildup. Since we don't have a lot of proteins that can create like that, like promote osmosis in our vessels, a lot of fluid just gets built up in areas based on gravity instead of our circulation. So kwashiorkor is a really, really interesting effect of protein malnutrition, right? Any questions? Oh yeah, just to be specific, at least know what these two things are and what's the difference. There's always a question on it as well. Any questions on this? So lecture's 15. So now we're going to start getting into the chemical side of biochemistry. Again, take it as you will. But we have one very important concept we need to know about amino acid, and it's just the mobilization of functional groups of amino acids. So in this case, we're going to talk about transaminase, which is just a transfer of the amine group between different amino acids. Okay. Remember, we have two versions of amino acids per se, or two different versions of the carbon skeleton of amino acids. We have our keto acids and our amino acids, right? So our transaminase is going to involve the transfer of the amine group from an amino acid to a keto acid. Okay. So you're going to see that we actually have two half reactions. We have the one that goes from amino acid to keto acid, which uses an enzyme called PLP. Then we have another reaction, which is a keto acid to amino acid that uses an enzyme called a PMP. Okay. And you're going to also notice that for the most part, these two half reactions are at equilibrium constant of 1. Okay. Can anybody tell me what that means? What an equilibrium constant of 1 means?
Was it the same bonding in the substrates and the products or something like that?
Well, it's like it's in a dynamic equilibrium, right? The products are matching the substrate, right? It's in that equilibrium phase. So that's actually pretty interesting as well, because depending on certain conditions of your body, that a constant is going to switch around, and it's going to mobilize to 4 more keto acids or 4 more amino acids, depending, right? So in cases of famine, we can actually create like ketone bodies, right? And the base of ketone bodies are actually keto acids as well. Okay, so these are like different components, right? But it's interesting to note that these two half reactions are in a dynamic equilibrium in a healthy human.
Wait, could you say that again? The four bit.
The 4
You
bit.
said.
Like the reason. I mean, basically, since we have this dynamic equilibrium in the amino acids, if there is any change in our body and our metabolism, it's going to affect this equilibrium as well, and is either going to move the reaction to make more amino acids or more keto acids, depending on the condition. So an example I gave is in extreme famine. We actually form certain things called ketone bodies, and for some of those ketone bodies can actually be formed from keto acids. So if we're in extreme famine, we're going to break apart the amino acids to form these keto acids to create like metabolic things to create more ATP.
Okay.
Yeah. And there's two transaminase reactions that we need to know, and it's our alpha ketoglutarate transaminase and our oxaloacetate transaminase. So here's the image, right? So you're going to see that we have our amino acid and then our alpha ketoglutarate, right? So our amino acid and our keto acid. And then we can actually form our glutamate, which is the amino acid of alpha ketoglutarate, or a pyruvate, which is the keto acid of alanine. Okay. So it's just switching around this amine group. Okay. So you see that the amine group of alanine is then transferred to the alpha ketoglutarate, and then this alpha ketoglutarate actually becomes glutamate. Okay. It's just a conversion of.
I have a quick question.
Yeah.
So that is representing the two half reactions that would utilize the PLP and PMP. He's just not
In this image, yeah, in this image, it's not showing those two half reactions that are occurring
like a
simultaneously.
sub reaction.
Yeah, but
Okay.
the big thing here, especially if you look at the arrows, you're gonna see that the arrows are the same size, which is just indicating that's in that dynamic equilibrium. It's that in that that equilibrium constant of 1, right?
Okay.
You-
So I have a quick question on that then. So back on the slide about the PLP and the PMP, and then it says the two half reactions. Is this a coupled like reaction that shows the two different directions it can go, or
It's basically showcasing that there's two enzymes that are working at the same time. We have our PLP and our PMP.
right?
For every time that a keto acid is formed from an amino acid using PLP, a PMP is converting a keto acid back into the amino acid.
So they're coupled together and they're
Closer
always
to-
going.
Opposite directions.
Okay. So each time one does one thing, the other one does the opposite.
Yeah, to maintain that equilibrium constant of 1.
Okay. All right. I just wanted to make sure I understood that concept.
Are they hooked up or is it just somewhere else in the cell a PMP is doing the opposite of the PLP?
I honestly don't know if they're like in close proximity or coupled up. I would say no, but they're probably in the same compartment of the cell. But I don't think they're coupled together. Yeah. But like that's a like that it's a good question, but he's not gonna ask like specifically that like if they're coupled or not. But I think like they would most likely be in the same compartment, so they would like feel the changes in the equilibrium constant. So just going back in, so we just talked about the alpha ketoglutarate transaminase. The oxaloacetate transaminase is that same concept, right? So we're converting glutamate to alpha ketoglutarate and oxaloacetate into aspartate. It's that same concept as well. So these two reactions are pretty important as well to know, right? So selectivity. So there's actually 4 amino acids that can't be transaminated. Okay, so out of the 20, there's 4 that can't. So proline can't, okay, because it has a secondary amine instead of primary amine. Hydroxyproline also can't because again, it's just a variation of proline. It's a secondary amine as well. Lysine actually, when it's transaminated, it actually cyclizes itself and creates a toxic metabolite. So we don't want that. And then threonine actually dimerizes, so binds with another threonine and actually becomes a toxic metabolite as well. Okay, so these are the 4 different amino acids that can't be transaminated. And you're gonna see right here, like with lysine, you see like as soon as it forms a keto acid, you're gonna see that we're actually gonna create a cyclization, right? It's pretty similar to like how we do with the glucose, right? It just attacks the carbonyl, and this becomes a really toxic metabolite. And for threonine, we just form a dimerization. So the 2 keto acid threonines are gonna actually bind to each other and going to create a toxic metabolite. So these are the 4 ones that we don't want to be transaminated, right? We don't want them to form a keto acid. Any questions on this?
Are we ever gonna find out why they're toxic, or is that just we just need to know they're toxic?
You just need to know, like, you don't need to know they're toxic. You just need to know that these are the 4 that can't.
Okay.
So now we're going to go into a new reaction, which is the glutamate dehydrogenase. So I'm just going to be straight up right now. These are the reactions that Dr. Pirich investigated during his doctorate work. So he knows these reactions really well when he used to work for the NIH. But so our glutamate dehydrogenase, right? So this remakes our alpha ketoglutarate and provides ammonia to be disposed. Okay, so we're actually removing our amine group to form alpha ketoglutarate. Okay, so we're converting our glutamate into its keto acid, which is alpha ketoglutarate. Okay, so this actually provides us with ammonia, and this ammonia is going to be disposed in the urea cycle, which we're going to talk about next week. Okay, so this is a really important reaction because it's going to involve itself with a huge metabolic pathway like next week. So this is a really big reaction that you should know all the components, right? So you're going to see that NAD plus is actually like assisting us in this first part of our reaction. So we're actually losing a hydrogen ion or hydride to our NAD plus to give us NADH and forming this amine, which is just an NH2 or an NH with a double bond. And afterwards, we can actually hydrolyze it. So we can actually use water to form a ketone and give us ammonia. Okay. And then again, ammonia is toxic for our body. So then we're going to dispose it through a different cycle. Okay. Any questions on this? Yeah, just know these components because this is a really important reaction. And then NADH and NADPH, right? So NADH is going to be the one that we're mainly going to be concerned about. And this is just mostly for the storage of electrons. So they're the transporters of electrons throughout our body. NADPH is pretty special because not only can it transport electrons, but it's mainly going to be used for biochemical synthesis. So when we're going to start talking about purine and pyrimidine synthesis, oxidative damage, they're going to be really important for synthetic pathways. Okay. Any questions on this slide? I know this reaction was a lot. Okay, so what drives glutamate dehydrogenase? So when we have high ATP, GTP, and NADH, GDH is actually going to be inhibited. Okay. When we actually have high ADP, high GDP, free amino acids, and NAD plus, this actually activates this pathway. Okay. And remember when I said that keto acids are pretty important for creating energy and ATP in different pathways, right? When we're talking a little bit about the importance of keto acids, alpha ketoglutarate is going to be really, really important for some of these pathways. Okay. So if we have high ATP, GTP, and NADH, this is just indicating that our body has a lot of energy, right? This is all indications that we have a lot of energy. So that's why it inhibits GDH, okay? Because we don't need to make more keto acids to make more energy because we have enough energy, okay? When we have a high amount of ADP, GDP, free amino acids, NAD and NAD plus, it's just indicating that we don't have enough energy anymore, and we need to start making keto acids to go through these pathways to make more ATP. Okay. So the way that you should always associate with inhibition and activation of pathways is, does my body need this at this point? Does my body need to break down this to make something else? If it doesn't, it's going to be inhibited, right? Because our body is constantly trying to stay in that homeostasis. So let me repeat what I just said. So in our GDH and our glutamate dehydrogenase, we form a keto acid in the form of alpha ketoglutarate. Alpha ketoglutarate is actually really, really important in metabolic pathways to form ATP and different energy molecules. Okay. So if we have high ATP, GTP, and NADH, these three molecules are indicators that we have a high amount of energy. So we're going to inhibit that GDH pathway because we don't need to form more keto acids to make energy, right? Because we have energy already. If we have high ADP, high GDP, free amino acids, and NAD plus, is an indication that we have low amounts of energy and we need to start activating that GDH just to form that keto acid. Okay. And then when will deamination occur? So deamination is when we completely remove an amine group as well, right? So in this case, this is a deamination because we're not transferring. So deamination is usually favored at low pHs, and high pH actually favors amination. So when we put that amine group back in, okay? I don't know exactly why, but this is for the information we got, right? I don't see like how detailed he's going to be with this. And then we do have something else called our L and D amino acid oxidases. Does anybody know the importance of these two enzymes? Okay. So which amino acid does our body use? The L version or the D version?
L.
Exactly. So we use our L version. So the amino acids aren't really going to help us that much. So basically, these oxidases are actually going to help us either think, yeah, break apart the D amino acids so that it's just not accumulating, right? Because we're not going to use it. So there are other deamination routes. We have our glutaminase, asparaginase, and our histidinase. They're going to follow a similar pathway to our GDH, to our glutamate dehydrogenase, okay? So for glutaminase, this is actually the hydrolysis of glutamine. So we're actually going to be using water, okay? This is present in the mitochondria. And this is pretty relevant because we're going to be forming these ammonias, and these ammonias are going to be really important to the urea cycle, which is associated with our mitochondria. That's why it's pretty important that it's in the mitochondria. Then we have asparaginase, which is just a hydrolysis of asparagine. We're going to also get ammonia, and then histidinase, which is the deamination of histidine. Okay, these are all reactions that you also should know, right? But again, they do follow a certain pathway, a similar pathway to GDH. Okay, but GDH is the only one that he wants you to know in full. Oh, this was actually my last slide. I thought there was a lot more, but okay. Any questions on lecture 15? Because I know that was like a lot of information right there too. And if you don't have a question, like we just ended too, so you're also free to go. I hope you guys have a great week because my week's been stressful this week. So you're free to go if you don't have any questions. I will be sticking around for like 10 minutes. So if anybody has any questions, just go free.
Can you go over the L and D like just one more time? 'Cause I feel like I
The oxidase. Okay. Yeah. So basically, like for humans, we only use the L conformation of amino acids, not the D version. So when we do have, like, for example, in dietary, sometimes we'll get an amino acid that has a D, but there's no use to it. So we usually have these to kind of scrap the D amino acid, if that makes sense. It's a very small detail in your slides. So it's basically a way that we can just prevent the amino acids from accumulating.
Okay, thank you.
Yeah, it's not huge. No worries.
Do we use the scraps to produce other things?
I mean, yeah, we could use the carbon skeleton to do any other things, but he doesn't want you to know what these other pathways are. The thing is too, like sometimes even our bodies make the amino acids by accident too. So our bodies ain't perfect. So we just need a way to kind of recycle or just trash them.
Is there a reason why PLP and PMP are kind of there? Like, why, if the cell decides to create a keto acid out of amino acid, it's just going to immediately have another one reversed? Like, it seems kind of counterproductive if.
I mean, like the way that I like to think about it is that both keto acid and amino acids are really important for our body, right? Because keto acids are important for energy and amino acids are important for protein building, right? So in a perfect situation, when our body is in like perfect harmony, these two reactions are in dynamic equilibrium, right? They're shifting from one to the other. If there's a huge issue where, let's say that we want to build a lot of proteins for some reason, one of those enzymes, which in this case would be our PLP, would actually start having some sort of inhibition that's going to prevent it from turning amino acids back into keto acids. But the PMP is going to still be working fine, converting keto acids to amino acids. So that way we have a huge accumulation of amino acids, which our body needs. So even though those reactions are working opposite, they're working completely opposite and perfectly opposite when we're in a perfect situation where we don't need one or the other more than the other, right? So
yeah, so basically they're kind of there for whenever the cell isn't doing anything, and it ensures equilibrium, but then when the cell wants to do something, it'll inhibit the one that'll like work against what it's trying to do.
Exactly.
All right, yeah, that makes sense now. Thank you.
No worries.
So would it be safe to say that those maintain equilibrium just based off of the concentrations within the cell? Like you said, they're probably in the same compartment. So
yeah, there's probably so many different signalings too, right? There's probably so much of your body does to signal if there is an increase of one or the need of another, right? So for example, like one thing that I can imagine, like these enzymes are probably have different reactions when insulin is present, right? Because insulin is indicating there's a high amount of like sugar, which also indicates there's a high amount of energy. And that's when we start creating proteins, when we're in that high energy state. So I could imagine that insulin would be there to activate the PMP or inhibit the PLP to allow more amino acids to be formed, right? Again, like there's probably so many signalings that are happening at the same time. But again, I wouldn't be able to name specifically all the signals if it's only based on concentration, which potentially, but the concentration is probably indicating something else, like a pH change or something else as well.
So you said that keto acids are important for energy. Could you like just go over again? How is that? Like, are they you producing ADP when you
Yeah,
convert?
so
Break 'em down.
basically, it's just because like what you're asking is all going to be exam 3 material basically, but I can just show you like something like right now of like, let's think. I'm trying to remember when that came out. So oxaloacetate, for example, right? Oxaloacetate, citric acid cycle. Yep. So you're gonna see that oxaloacetate. Can you see my screen? I don't know if is it good? Okay.
Mhm.
So you're gonna see that oxaloacetate is present right here. Okay. And this is a huge cycle that you're gonna learn in exam 3. It's not an exam 2. I'm just for reference. Oxaloacetate is very important for this cycle right here. And this is going to form a lot of our NADHs and FADHs, which is eventually going to form a lot of ATP. It's going to form around 30 to 32 in one cycle. Okay. So you're going to see like this is one example of a keto acid that's really important in a certain like energy metabolic pathway. And a lot of these keto acids are pretty important. I just, for some reason, I know alpha ketoglutarate is important. I just don't remember which pathway it is for some reason. But alpha ketoglutarate is pretty important in another energy metabolic pathway as well. Actually, yeah, it was this one. Sorry. See, alpha ketoglutarate is right here as well. So oxaloacetate and alpha ketoglutarate are both very important for this one reaction that makes a lot of ATP.
The good news about that is you can go and put that on a shirt.
Yeah, so keto acids are like they're very important in creating energies, and proteins and amino acids are very important in creating proteins. So they really, in general, they're both have separate functions, but they're both very, very important in keeping us healthy, right?
Okay. All right, thanks. Yeah, I wasn't sure if I had like missed the point on there where you said, oh, it's creating ATP at this step or something.
Oh no, no, that's all exam 3 materials. That's all gonna come eventually. Any other questions?
I'm gonna drop off. Thank you very much.
Yeah, no worries. Have a good night.
Do you have any advice on how to like go about studying this in any kind of order? I mean, somebody had shared a diagram she's made, and I might try to create one myself just because that will help me. But I just feel like all this information is just floating out there, and I would do so much better with like some form of flow chart where I could be like, okay, here's this stuff.
Yeah.
Stuff.
So right now it still hasn't been like reaction heavy, but like the next couple of weeks is going to be a lot of reactions that are kind of connected to each other.
Okay.
They're not as much as they could be, but there's just a lot of reactions. So that's why that worksheet that I gave is like pretty good just to have everything in one paper.
Okay.
But I think the way that I studied for this, I think it was just flashcards because there was just so many reactions that I just needed to like actively try to recall every step of the reaction.
Okay. I just feel like there's all these different puzzle pieces floating out there and they don't quite fit together.
Yeah, no, like it's just because it's the beginning of biochemistry. Like it's hard to connect when there's not a basis yet, if that makes sense.
Yeah, and that's what I'm trying to. My brain works really well when I can organize things in a more
Yeah, like-
linear fashion rather than having it all coming from all these different angles. So
No, I completely get you. I'm the same way.
I'm always looking for patterns. I'm like, how does this go?
I think the one thing I had to learn in college is that sometimes not everything has patterns, and you just have to know. Just-
Yeah. I like when there's a rule and I can count on that rule every time. I do that in my exam all the time. I'd be like, so is that the rule every time? And the teacher would be like, eh.
It's like for the class, sure. Any other question?
Nope, I think I'm gonna go, but hopefully it'll start coming together. I just need to start memorizing, I think.
Yep. Okay. Have a good night.
All right, thanks.
Just gonna end, pause the, stop the recording.